So last time we were talking about how DNA is coiled up into chromosomes. And if you were to unwind a section of DNA, it's actually two separate strands wound around each other like a twisted ladder called a double helix. Today we're going to unwind that and look at one of those strands. And then we'll add the other strand back in. So keep in mind that DNA is actually two strands wrapped around each other, and then that is really the DNA molecule that's then wound up like a phone cord. We're going to unwind it, look at one of them. So if we look at one of them, you will see on the screen, you've got a series of these circles, pentagons, and rectangles. And one of those is a unit called a nucleotide. And this picture has 1, 2, 3 nucleotides. That represents one of those strands. On the other side over here would be another one of them, and they would be twisted together. But again, we're just going to look at the one, just the one strand for the moment. If you look, we've got the one strand, that series of nucleotides, and then we've got another series of nucleotides on the other side like that. They're attached loosely by something called weak hydrogen bonds. And they will hold it together so that it can unzip and zip back up. And as you will see, that is part of the, serves part of its function. The fact that it can unzip and zip back up is really important to its job of making us look the way we do. Recall that I said I can't explain that to you until we understand the structure better. So we'll hold off on that. Just know it's really important that it be able to unzip and zip back up. All right, so that's unzipped one single strand we're going to look at is made up of a series of nucleotides. This is one of them. And we had the circle, the pentagon, and the rectangle. The circle is a phosphate group. The pentagon is a sugar, a ring, right? A sugar called deoxyribose. And the rectangle is a nitrogenous base. Nitrogenous is a fancy way of saying it has nitrogen in it. Like remember, we had carbon, hydrogen, oxygen. Well, nitrogen is in this. So this is a nitrogen base. And that's what those shapes represent. Now, you don't need to know this, but the circle, the phosphate, actually represents all those atoms hooked together. And the pentagon, the rectangle of the pentagon, the sugar, deoxyribose, actually represents all those atoms hooked together. And the rectangle, the base, represents all those. So every time you see all this drawn with the circle, the pentagon, and the rectangle, we really mean a whole mess of atoms. And remember that those are all hooked together in a long chain with another chain of them wrapped together. So this is a giant molecule, giant, with tons of atoms. Now, remember that those nucleotides, the circle, the pentagon, and the base, are hooked together in a long chain. Well, there are 4 different kinds that can be hooked together in that chain. One is A, another is G, a third is C, and the fourth is T. You might see on a test the words adenine, guanine, cytosine, and thymine, but you only need to identify them as A, G, C, and T. So you'll never have to say adenine. You can just say A, even on the Regent switch. You don't have to take. So you have this series of nucleotides all down the strand. And then on the other side, you have a bunch of nucleotides all down the strand. And they're hooked together by those loose hydrogen bonds that weakly allows them to zip up and unzip. So the order of bases on one determines the order of bases on the other, because if there's an A over on this side, it means there's a T over on this side. If there's a C here, there's a G there. So A always matches with T and C always pairs with G. So if we take a look at this strand right here, we have a phosphate, a sugar, deoxyribose, and a nitrogenous base. This time it's T. That is hooked together with that nucleotide, which is hooked together with that nucleotide. That all makes one strand. The other side, one nucleotide, another one, and another one makes the other strand. Those two are held together by these loose hydrogen bonds, and then that whole thing's twisted together. Now, if there's T on this side, what must be over here? Well, T always matches with A, so there'll be an A here. If this is A always matches with T, so there'll be a T here. C matches with G. We say that the order of bases on one side is complementary to the bases on the other. So T matches with A matches with T, C matches with G. Here in this picture, we have 1, 2, 3 nucleotides on this side. And on this side, we have 1, 2, 3 nucleotides. This strand of nucleotides are complementary to that one. Now, it gets pretty old drawing circles, pentagons, and rectangles. So we can just use a line to represent the phosphates and sugars, the circles and pentagons, because they never change. Every single DNA, it's just phosphate, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose. So we can just represent that with a line. Same thing on this side, deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate, a line. The only things that are different from one nucleotide to the next are the bases on the inside. So we can simply draw the letters and stop representing the circles, pentagons, and bases all together like that. So what we really have here is a DNA molecule where truly the circle is really there, the pentagon is there, and the rectangle is that. So we have 1 nucleotide, 2, 3, 4, 5, 6. So what would be complementary in each case here? We'll do the first one together. A matches T. T matches A. C matches G. G to C. G to C. G to C. C to G. A to T. T to A. And A, T, T, T.